This is Physiology lecture number 10 in the respiratory module. This lecture explains how the higher center control or regulate ventilation. Lecture objectives: describe the central control of ventilation, identify the sensors, various chemoreceptors that provide the respiratory center with information, describe the integrated responses to carbon dioxide, hypoxia, and the pH, discuss the regulation of ventilation in chronic respiratory failure. The respiratory center. The respiratory center is a group of neuronal collections within the medulla oblongata and the pons. The function of the respiratory center is to adjust alveolar ventilation to meet the demands so the partial pressure of oxygen and carbon dioxide in the arterial blood are held constant even during exercise. Physiological anatomy. The respiratory center is composed of 4 groups of neurons located bilaterally in the medulla oblongata and the pons. These groups are: first, the dorsal respiratory group. This group is responsible for the initiation of inspiration. The ventral respiratory group. This group causes mainly expiration. The pneumotaxic center. This center controls the rate and depth of breathing. We will understand later how is that. This group is located dorsally in the superior portion of the pons. The last group of the collection is the apneustic center. The apneustic center really is in the inferior or the lower portion of the pons. Its function in the human being is questionable. There is an important note I would like you to note all. The pons, the hypothalamus, the reticular activating system, and the cerebral cortex, as well as efferents from the vagus, glossopharyngeals, and somatic nerves, influences both the dorsal and ventral group of the respiration. This diagram shows you the organization of the respiratory center. Please note that the pneumotaxic center is present in the pons, while the dorsal respiratory group and the ventral respiratory group, both of them are present in the medulla. Also note please that efferent fibers from the vagus and the glossopharyngeals are just for the dorsal respiratory group. The ventral respiratory group receives no efferents from the spinal cord. However, both dorsal respiratory group and ventral respiratory group, they send efferent fibers down to the diaphragm and other inspiratory and expiratory muscles through the respiratory motor pathways. Now let's understand the function of the dorsal respiratory group. Neurons of the dorsal respiratory group are almost located within the nucleus of the tractus solitarius. The neurons extend most of the length of the medulla. Please remember that the nucleus of the tractus solitarius is the terminal station for the sensory part of the vagus and the glossopharyngeal nerves. From the chemoreceptors, from the baroreceptors, and other receptors in the lungs. In fact, the dorsal respiratory group is the pacemaker for the basic respiratory rhythm. This group of neurons can operate and transmit impulses, and they don't need any impulse from the higher centers such as the cerebral cortex or any other higher centers, or from lower centers, that's to say impulses from the spinal cord through the vagus or the glossopharyngeal to function. They are the pacemaker, and they can function independent of all other centers. However, other centers can modify and affect the function of the dorsal respiratory group. The output from this group is transmitted to the primary inspiratory muscles through the phrenic nerve to the diaphragm in a ramp or a crescendo manner. Let me explain to you what is meant by ramp or a crescendo manner. It means simply that this center sends first impulses in low frequency, and with the time the frequency will increase little by little, and this will make the inspiration smooth. It will start simply by simple intake of air, and then the intake will be more and more. By such, there will be no gasp. Let me make it as a sound. Simple inspiration will be like this. But if the impulses comes in on one burst, very high frequency, it will be like a gasp, like this. This will not happen, simply because the ramp of or the crescendo fashion is smooth. Now, the longer the interval of time for the ramp means the span will be wide, the deeper will be the inspiration, and greater will be the tidal volume. So remember, the ramp has a rate and a limit. Limit means a span that can be controlled. The greater the increase in the rate, the greater is the filling of the lungs, means the greater will be the tidal volume. Whereas the shorter the limit, means shorter is the respiratory cycle, and the higher will be the respiratory rate. The pneumotaxic center. This center is located dorsally in the nucleus parabrachialis of the upper pons. The main function of the pneumotaxic center is simply to switch off the function of the dorsal respiratory group. So the switch off point of the inspiratory ramp in the dorsal respiratory group will control the tidal volume. Let me explain this further. As I told you, the size of the tidal volume depends on how long it takes for the ramp. So if the span increases, simply now the ramp is going on and on, the pneumotaxic center is designed to switch off the ramp when the tidal volume reaches to about 500 mils. And when switch off of the cycle takes place, expiration will follow, and a new cycle later will begin from the dorsal respiratory group. Now let's imagine this. If the switch off of the pneumotaxic center is delayed, means the tidal volume will get bigger, and the inspiration will take longer time. And when the inspiration takes longer time, also the expiration will take longer time. This means the respiratory cycle will be slow, and we will have a slower respiratory rate. On the contrary, if the switch off of the pneumotaxic center comes early, this means the tidal volume will be smaller, will be less than 500 mils. The inspiratory time will be shorter. The expiratory time will be shorter as well, and the respiratory cycle will be faster than usual. So the pneumotaxic center simply it controls the volume, and this will inversely related to the respiratory rate. How powerful is the pneumotaxic center? It can send very powerful inhibitory effect to the dorsal respiratory group. Strong stimulation of the pneumotaxic center can shorten inspiration for as little as 0.5 or half a second. Remember, the inspiration takes about 2 to 2.5 seconds. The function of the pneumotaxic center is primarily to limit inspiration, leading to a shallower and more rapid respiratory pattern. The ventral respiratory group. Neurons of this group are located in the nucleus ambiguous and the nucleus retroambiguous. The ventral respiratory group remains almost totally inactive during normal quiet respiration. Physiologists, when they examined this group of neurons, they found no electrical activity can be detected from this group during normal quiet respiration. Therefore, the ventral respiratory neurons do not appear to participate in the basic rhythmical oscillation that controls respiration. Definitely now you will ask, what's the function of this group now? This group helps during expiration. When the respiratory rate increases, such as during exercise, this starts by hyperactivity in the pneumotaxic center or pneumotaxic group of neurons. The increase in the activity of the pneumotaxic neurons, some of the activities will spill over to the ventral respiratory group and activate them. And the superactivation of the dorsal respiratory group also will activate the ventral group, and now the ventral group will start to operate to help and to assist the dorsal group when a greater pulmonary ventilation is needed. It will help in the expiration function. The ventral respiratory group supplies the expiratory signals to the abdominal muscle during very heavy expiration. The apneustic center. This center is located in the lower part of the bones. In experimental animals, please, I will repeat this. In experimental animals, it inhibits the switch off of the inspiratory ramp signal. That will lead to an increase in the tidal volume and duration of inspiration with occasional expiratory gasps. That's why it is called apneustic center. Apnosis means deep inspiration, deep continuous inspiration, as if you want to make inspiration endless, but of course it will never be endless. After such huge value for the inspiration will be switched off for short interval and it will be repeated again. This is what is called by apnosis. This is discovered in experimental animal. When they make a cut in the bones between the apneustic center and the pneumotaxic center, as such they terminate or they make the when you make the cut above the apneustic center, means you cut the neurons coming from the pneumotaxic center to the dorsal respiratory group. And the dorsal respiratory group will be just under the control of the apneustic center, and that's what will happen to the respiration. Its activity is inhibited. The activity of the apneustic center is inhibited normally by the pneumotaxic center and by vagal impulses. What's the role of this center in a human being is questionable. We don't know what. It has only discovered in experimental animals. Anyhow, maybe in the future the scientist will discover a function or another function for this center. The Hering-Breuer Inflation Reflex, or the inspiratory inhibitory reflex. This reflex is named after the name of two German scientists, Joseph Breuer and Edward Hering. This reflex simply is a reflex triggered to prevent the overinflation of the lung. The stimulation of the bronchial and bronchial stretch receptor by the overinflation simply will induce an efferent signal through the vagus, and this signal will end in inhibition of the dorsal respiratory group and the apneustic center. As to say, its function is similar to the function of the pneumotaxic center. As if the pneumotaxic center does not work for any reason, the inflation of the lung should be terminated at a certain point. So when the inflation is going on and on by the ramp, when the size of the tidal volume exceeds 1.5 liters, right away the reflex will operate and terminate the inspiration and the overfilling of the lung. And this will allow, this inhibition of inspiration will allow the expiration to occur again. Please remember that 1.5 liters tidal volume, that will be 3 times the normal value. So this reflex simply is a protective mechanism. The reticular activating system. We said in the previous slides that the dorsal group can be affected by other centers in the central nervous system, like the cerebral cortex, like the hypothalamus, and also it will be affected by the reticular activating system. You student, maybe at this level of your physiological teaching, you have not gone in depth with the function of the reticular activating system. You will know that when you will have the modules of the central nervous system. Let me give you a few words about it. The reticular activating system simply is a group of neuronal connection present in the medulla oblongata and in the pons. The function of this system is to keep us awake. When we are sleeping, this system will be inactive. Once this system is activated, we will wake up from the sleep. And for this reason, the reticular activating system can have an effect on the dorsal respiratory group. The reticular activating system stimulates the respiratory center to increase respiratory drive. That's when we are awake. However, during sleep, when the reticular activating system activity is diminished, there is a decrease in alveolar ventilation that leads to a slight elevation of arterial carbon dioxide tension. And remember, when we are asleep, our heart rate will be the lowest, and also our respiratory rate will be slower. Up to this point of the lecture, we discussed the basic respiratory rhythm, thus to say, the respiratory rate when we are at rest. However, the respiratory rate changes during many physiological and non-physiological or pathological conditions. The respiratory rate increases during exercise. The respiratory rate increases during stressful conditions, such as when we are in pain or when we are excited. The respiratory rate stops altogether when we swallow food, and many other physiological and non-physiological cases. So we need to understand how can the respiratory center control respiration in different physiological and stressful conditions. The intensity of the respiratory control signals is increased or decreased to match the ventilatory needs of the body in many physiological and pathological states, such as exercise, hypoxia, and metabolic acidosis or alkalosis, and etc. The control mechanisms can be divided into two groups. These are the chemical control of respiration and non-chemical or the neural control of respiration. The chemical control of respiration is the most important one, and let's start with this. The chemical control of respiration relies on the respiratory receptors or the chemoreceptors. Scientists discovered that there are two groups of chemoreceptors in our body. The first group is the central chemoreceptors, and the second group is the peripheral chemoreceptors. The central chemoreceptors are chemosensitive neurons located bilaterally and are lying 0.2 millimeters beneath the ventral surface of the medulla. The neurons there are sensitive to changes in either the partial pressure of carbon dioxide or hydrogen ion concentration in the surrounding interstitial fluid. Please note that the interstitial fluid there is the cerebrospinal fluid. And any change in the partial pressure of carbon dioxide or hydrogen ion concentration of the cerebrospinal fluid will end in stimulation of the respiratory center. The second group of the chemoreceptors are the peripheral chemoreceptors. The peripheral chemoreceptors, as we mentioned in the lectures of the cardiovascular module, are located in the carotid and aortic bodies. They are merely the carotid body and the aortic body, they detect changes in oxygen in particular in the arterial blood, and also respond to a lesser degree to changes in carbon dioxide and hydrogen ion concentrations. Signals from the carotid and aortic bodies are transmitted through the glossopharyngeal nerve to the dorsal respiratory area of the medulla. The aortic body signals are sent through the vagi also to the dorsal respiratory area. The central chemoreceptors. The central chemoreceptors are stimulated exclusively by the hydrogen ion in the CSF. The hydrogen ion of the CSF are believed to be the only direct stimulus to the central receptors. Changes in the hydrogen ion concentration in the blood have less stimulatory effect, as hydrogen ion does not easily cross the blood-brain barrier. What this point means is that the hydrogen ion that stimulates the central chemoreceptors is produced locally into the CSF. It is not from the hydrogen ion in the peripheral circulation. Metabolic acidosis cannot affect usually the central chemoreceptors because the hydrogen ion in the plasma cannot cross easily the blood-brain barrier to reach to the CSF. So the local production of the hydrogen ion comes from the carbon dioxide reaction with water. So the effect of the carbon dioxide is indirect. Elevation of the carbon dioxide in the peripheral blood simply will reach to the CSF because the carbon dioxide can easily cross the blood-brain barrier. Carbon dioxide reacts with water in the CSF to produce the carbonic acid, and then the carbonic acid will dissociate into the hydrogen ion and the bicarbonate ion. The hydrogen ion then stimulates the central chemoreceptors. Once the central chemoreceptors is stimulated, this will increase the rate and depth of respiration. This is clearly demonstrated in this diagram. This diagram explains the effect of partial pressure of carbon dioxide in the peripheral blood and the effect of the hydrogen ion concentration, or the pH, of the peripheral blood on the ventilation of the alveoli. It is quite clear here from this graph that at a partial pressure of 40, which is the partial pressure of carbon dioxide in the arterial blood, once it increases from this value up to 45 or to 50, there is a tremendous increase in the alveolar ventilation, which means a powerful effect on the chemoreceptors. Whereas very minimal effect for the change on the pH can be attributed to the very minimal change in the alveolar ventilation can be attributed to the change in the pH. So this demonstrates clearly that the acidosis, the metabolic acidosis, when the hydrogen ion is in the peripheral blood, it cannot affect the chemoreceptors in the brain, while the carbon dioxide can do that. So any increase in the partial pressure of carbon dioxide increases the strength of both the inspiratory and expiratory motor signals to the respiratory muscle. Now comes a very important physiological question. Is such kind of receptors adaptable? The answer is yes. Initially, when there is an increase in carbon dioxide, the response for the increase in alveolar ventilation is so acute. It's so acute and it can increase it to a tremendous high levels. However, if the rise in the partial pressure of carbon dioxide in the peripheral blood persists, after 2 to 3 days, this response on the alveolar ventilation will start to decline and to decrease. So the chemoreceptors has a weak chronic effect on changing the respiratory rate and depth of respiration. Is this entirely because of adaptation? The answer is it is partial, not entirely. What happens within 2 to 3 days, there will be a kidney function other than the respiratory system to correct the acidosis. So if there is an increase in the carbon dioxide in the peripheral blood, This means there is acidosis, and the kidney will start to produce extra amount of bicarbonate to neutralize the hydrogen ion. So part of the adaptation is because of the kidney function, and the other part of the adaptation is because of the adaptation of the central chemoreceptors. The student should know that approximately 85% of the resting chemical drive of respiration results from the stimulatory effect of carbon dioxide on the central chemoreceptor. If we expose the central chemoreceptors to low level of oxygen, that's to say hypoxia, the central chemoreceptors will not be affected. So oxygen changes have no direct effect on central chemoreceptors. Therefore, and this is a very important point for you as a future doctors, you should know that carbon dioxide is the major controller of respiration and not oxygen. The peripheral chemoreceptors. As we mentioned before, central chemoreceptors are not sensitive for changes in oxygen concentration in the peripheral blood. So how can the respiratory centers detect changes in the oxygen? Simply, they get the information through the peripheral chemoreceptors. So the peripheral chemoreceptors are the only site that detect changes in oxygen in the arterial blood. They respond to a lesser extent to changes in carbon dioxide and hydrogen ion. I would like the student to remember what we have said before about the oxygen dissociation curve. When we discussed the curve, we mentioned that the saturation of the hemoglobin is almost complete between the value of 70 millimeter mercury up to 97 millimeter mercury, and the real decline in the saturation of the hemoglobin with oxygen begins below the value of 70 millimeter mercury. And here we notice that the peripheral chemoreceptors have been designed to do this. That's to say, they start to detect the change in oxygen concentration changes when it goes below 70 millimeter mercury. So, a decrease in partial pressure of oxygen below 70 millimeter of mercury will end in stimulation of the peripheral chemoreceptors, and the stimulation of the peripheral chemoreceptors results in stimulation of the dorsal respiratory group and increased rate and depth of respiration. That's to say, hypoxia can cause an increase in rate and depth of respiration. What about the carbon dioxide and the hydrogen ion in the peripheral blood? The answer is an increase in either carbon dioxide or hydrogen ion concentration. Both will stimulate the peripheral chemoreceptors, and this will end in rapid stimulation of the respiratory center. This stimulation is rapid, much 5 times before the stimulation can be attributed to the central chemoreceptors, meaning the peripheral chemoreceptors will detect earlier the changes in the carbon dioxide and in the hydrogen ion concentration, and simply they act as a sort of feedback, a feed forward for the central nervous system to deal with the rise in carbon dioxide and hydrogen ion concentration. The diagram here shows you the site of the peripheral chemoreceptors. As you can see clearly, it is in the carotid body at the site of the carotid sinus and in the aortic body. Here in this diagram, it shows you that the increase in ventilatory drive due to hypoxia is multiplied if central chemoreceptors loses its sensitivity because of the adaptation. What does that mean? If somebody goes high up into a mountain, and as we know that at high altitudes the oxygen concentration is much less, so the peripheral chemoreceptors will simply be stimulated, and this should stimulate respiration. But the respiration will not be changed greatly initially, for very simple reason. When there is an increase in the respiration, that means there is an increase in the washout of carbon dioxide from the body. And this will decrease the level of the carbon dioxide at the central chemoreceptors. And when the carbon dioxide will decrease at the site of the central chemoreceptors, this will not act in favor of increasing the respiratory rate. In fact, this will dampen or try to decrease the effect or counteract the effect of the peripheral chemoreceptors. So the increase in the ventilation will be minimal once we go up into the mountain. But after 2 to 3 days from ascending into a high mountain, little by little our respiration will start to be better, and we will get deeper effect for the hypoxia, and the tidal volume will start to increase. And the scientific explanation for that is that the central chemoreceptors now have been adapted, and it will accept a lower level of carbon dioxide. So the effect of hypoxia will be greater. This is the term used to explain acclimatization, which is deeper breathing 2 to 3 days after slow ascending of a mountain. And please note that anemia, that's to say low level of hemoglobin, and carbon monoxide poisoning do not stimulate peripheral chemoreceptors. Respiratory regulation during exercise. During exercise, there is a tremendous increase in oxygen consumption and in carbon dioxide formation. The rise can be as high as 20 folds. Yet, in a healthy athlete, alveolar ventilation ordinarily increases almost exactly in step with the increased level of oxygen metabolism. During exercise, the partial pressure of oxygen, the partial pressure of carbon dioxide, and the hydrogen ion concentration, or the pH, remain almost exactly normal. So the bigger question is this: if there is no change in the partial pressure of oxygen and there is no change in the partial pressure of carbon dioxide or the hydrogen ion, means there is no stimulation neither for the central chemoreceptor nor for the peripheral chemoreceptor. So what causes the high level of increase in ventilation and the high respiratory drive in the respiratory center? So the immediate increase should be attributed to something else, and this is believed by most physiologists that the stimulatory impulses from higher centers such as the cerebral cortex of the brain to both the respiratory muscle and respiratory center is the cause. And this have some proof, because in well-trained athletes the increase in respiration is much better than in junior athletes or those who are doing the exercise for the first time. Means when the brain is anticipating doing the exercise, the increase in the respiration is better. So definitely the cerebral cortex has an effect. And the other factor that could be attributed to the change is the proprioceptive stimulatory reflex. And what we mean by proprioceptive stimulatory reflex is the reflex initiated from changing the angles of the joints and changing the degree of tone in skeletal muscle. The reflex initiated from these two sides can be attributed to the stimulation of the respiratory center. The non-chemical factors or neural factors that affect respiration are many, and I will give you here 5 examples. First, the voluntary control of respiration means when we try to stop voluntarily our respiration or increase voluntarily our respiration. The nervous pathway passes directly from the cerebral cortex and other higher centers through the corticospinal tract. Another example is the neural effect because of coughing, sneezing, and even breath holding, because that's to say apnea, which is due to stimulation of pulmonary irritant receptors. In viral infection or bacterial infection of the lung, there will be irritation to some receptors in the bronchi, or if we smell something which is quite irritant to the receptors also in the bronchus of the lung, these receptors will be affected and will send an impulse, and the response to this reflex can also cause a mucus secretion and bronchospasm, as in asthma and in emphysema. Other example, the feeling of dyspnea, which is difficulty to breathe, when the pulmonary capillaries become engorged with blood. So if the capillaries around the alveoli are engorged in blood, receptors will be stimulated. These receptors are called juxta receptors in the wall of the alveolus, and they abbreviate the name from juxta to J. They call it J receptors. They are present in the wall of the alveoli. Also, these receptors can be stimulated in pulmonary edema. So if these receptors are stimulated, the person will sense the feeling of dyspnea and stop of respiration, and their stimulation usually should end in tachypnea for correction of the feeling of suffocation. Also, the respiratory center can be affected, and this is very well known in pharmacology, by anesthetics. Anesthesia can cause respiratory depression and respiratory arrest in overdosage. And if you hear that somebody died because of anesthesia during surgery, this is because the anesthetic simply stops the function of the respiratory center. And the other example is the one we just said before, the inflation reflex, which is the Hering-Breuer reflex. It is also an example of a reflex that can affect the respiration to prevent overinflation of the lung. And remember, the respiration will stop by a reflex from the pharynx once we try to swallow anything, And there is many other examples. The last topic to be covered in this lecture is the chronic respiratory failure. Chronic respiratory failure is a syndrome in which the respiratory system fails in one or both of its gas exchange functions. That's to say, there is failure in the oxygenation and carbon dioxide elimination. Chronic respiratory failure is classified as either hypoxemic or hypercapnic respiratory failure. The failure may be complicated because of the chronicity by polycythemia or call pulmonale. Hypoxemic respiratory failure is characterized by an arterial oxygen tension lower than 60 mm mercury with a normal or low arterial carbon dioxide tension. We have known before that it is easier for the lung to get rid of the carbon dioxide, but usually if there is something wrong in the lung, the difficulty will appear with getting oxygen out of the alveoli to the blood. So hypoxia usually appears before hypercapnia. This sort of chronic respiratory failure is the most common form of respiratory failure. If the case is more bad, hypercapnic respiratory failure is characterized by a partial pressure of carbon dioxide higher than 50 millimeter mercury. Hypoxemia is common in patients with hypercapnic respiratory failure who are breathing room air. The patients with chronic respiratory failure and hypercapnic, usually they have respiratory acidosis. And the cause of the respiratory acidosis is simply because of either one, generalized hypoventilation and results from failure of the controller to respond to carbon dioxide. Thus to say, during anesthesia, following a brain injury, and in some patients with chronic obstructive lung disease. And the other cause of acidosis, if the problem is not with the respiratory center, the problem here is because of obstructive lung disease. When these patients breathe room air, hypercapnia caused by a reduced alveolar ventilation, which is accompanied by significant hypoxia and acidosis. As usual, if you understand the topics of this lecture, you should be able to answer this question. Again, if you have any difficulty with understanding or solving this question, please contact me and I will help you. Thank you very much and good luck.